Bio 181, Chapter 14 on DNA, and we're going to dive into the specifics and the molecular enzymatic processes that are required to create a newly synthesized strand of DNA. So DNA replication, that just means we're making a copy of the DNA. It occurs in the 5' to 3' direction. It requires a template strand. So we need a strand with some base pairs hooked onto it to make a new strand. And we are going to need some nucleotides. So there are nucleotides that are kind of floating around in the general vicinity, paired with different bases. So you might have, you'll have a nucleotide, some A's hooked to your, I'm trying to draw it backwards now. I can't do it very well. So I usually draw it like this, right? Here's my nucleotides. So I might have some bases with adenine. I'll have some bases with my cytosine hooked up to my sugar, hooked up to my phosphate. So those are floating around. We also would have guanine hooked up to my sugar, my ribose, and my phosphate. And then the last one floating around would be my, what am I missing? Thymine, right? My last nitrogenous base hooked up to my sugar and my phosphate, okay? So these are what we'll need, plus some enzymes to help put them together. So I've got my building blocks, my monomers, my 4 possible nucleotides, 1, 2, 3, and 4. And then we're going to put them together. So the enzyme sees that cytosine is there. It's going to match that with the guanine, right? And then the guanine is going to be matched with the cytosine. It's going to catalyze the formation of hydrogen bonds, and you're going to have the elongation of the newly synthesized strand. So we're going to specifically look at multiple enzymes. One is called DNA polymerase. ASE on the end indicates it's an enzyme. It's making DNA into a polymer. So remember, if you put a lot of monomers together, you've got a whole bunch of building blocks, you link them together with covalent bonds into a really long molecule. That molecule is called a polymer. Right? So polymers are made up of monomers. So DNA polymerase is a big enzyme that's going to catalyze this reaction. A couple other enzymes that you'll be responsible for are helicase, again, ASE on the end, primase, and ligase, all indicating that they are enzymes involved in the process. Okay, so I decided I would just have this one kind of busy slide and take a little bit of time to go into it. You will be asked, you may be asked to label a very similar diagram, if not this diagram, with the names removed. In addition, I'll ask you potentially to know the function of each of these. So let's just orient ourselves. So we've got this parent strand of DNA over here on the left. Okay, so here's one of the sides with the bases that would be linked to this anti-parallel side that's on the top. Okay, and again, they're linked together with hydrogen bonds. So if we zoomed in, we would see that in between each of these bases, there are some hydrogen bonds that are holding them together fairly weakly compared to covalent bonds, right? So we're going to break the hydrogen bonds. Helicase is the enzyme that is going to unwind this helix. So it unwinds the spiral structure or the helix spiral. Okay, and that's not really shown here, but this starts off as a structure that's like a spiral staircase, and so it's winding around. And helicase, which is this enzyme right here, is going to help unwind that. So taking it from a spiral staircase into just a straight staircase. So that's really the function of helicase. Once that happens, so it's separating, unwinding these two and helping to separate them, then we are going to actually stabilize the separated strands by adding in these single-stranded binding proteins, and they're blue. So I'm moving down here. This isn't actually an enzyme, but these are going to be placed periodically on the open strand so that they don't bond again, right? Those base pairs want to bond together, so those hydrogen bonds are broken, but to keep them from going back, we kind of put these bumpers on them so that they won't bond or re-anneal. And so they abbreviated SSBPs. These are single stranded binding proteins. And that's what these are. So all of these. Okay. All right. So once we open it up, then we have the enzyme primase. So primase, let's see here, it's kind of in a purple color, I guess, more like a liver red, right? But this enzyme here, primase. So primase is the first enzyme that's going to be adding nucleotides. So it's going to actually add RNA nucleotides. And we haven't really talked about that nucleotides. But there are some key differences between RNA and DNA. One is the sugar. Yep, one is the sugar, which is ribose. One is the base, which they replace thymine with uracil. And then the last one is the fact that it makes single-stranded versus double-stranded molecules. All right, so RNA, got a little distracted there, sorry. So RNA is going to be added here. So these first nucleotides that are added are going to be RNA. So you'll see in these base pairs some uracil, right, along with like that maybe the adenine and the guanine, but this is RNA. So all of these little initial segments that are shown here are what are called RNA primers. So that's how it has to start to make the DNA molecule. After primase lays down or adds on the RNA nucleotides, then DNA polymerase can come along and actually add in the DNA. So once the RNA has been synthesized, it can hop on after a few base pairs are made. So say down here is where it started. And then all of these molecules here in blue are going to be DNA, right? So this would be your T's, C's, G's, A, C, T. So there's no more uracil. It's not RNA anymore. So this is my DNA strand. So the molecule that's doing that, the enzyme, is DNA polymerase. So we've got DNA polymerase, and it's going to continue until it bumps into the segment that's already been synthesized, or if we're up here at the top, until it runs into the segment that's just open. Ligase, so the last enzyme that we'll talk about here is shown here down at the bottom right, and ligase is going to catalyze the bond between these fragments that are down here. So there's little pieces that aren't catalyzed, and so it's going to catalyze this covalent bond. So ligase is going to link together these fragments. Okay. And so what are these fragments? Why is it not continuously synthesizing? Well, on one side it's discontinuous, and on one side it's continuous, and that's because it can only synthesize, we go back here, whoops, in the 5' to 3' direction. Okay. So this results in a leading strand, which is going to be continuous, and a lagging strand, which is going to be discontinuous. So up here on the top, this leading strand, DNA polymerase can just keep going straight, no problem, right? Just adds, oops, it's down here. It's adding on new nucleotides. But on the other side, since it can only go in one direction, it has to hop on and move away from that opening. And so it starts synthesizing bases, but then the section that is ahead of it opens up and it needs to jump on again. So you see these segments that are fragments, and these fragments are called Okazaki fragments. So this is the name of a Japanese married couple scientists that initially identified these. And ligase is the enzyme that will link these together. All right, so that's it for the enzymatic structure of DNA replication.